Hello, in this video I'll show how to collect saliva samples. So we provide these tubes, collection tubes, with a small pipette. So basically you roll your tongue inside your mouth to collect the spit, your saliva, and dispose it into the tube. And then close the cap, shake it to move everything down the tube. So this is enough saliva collected for the next step of the Proteinase K treatment. At this phase, once we have collected the saliva sample in the tube, now we are going to transfer it into a standard PCR tube. So I'm going to open the cap here. We have a standard PCR tube. So we will use a pipette that is already adjusted to 200 microliters. I will be transferring 200 microliters from the larger tube into the smaller one. But 200 microliters. And then the next step is to transfer the Proteinase K . 26 microliters. Into the PCR tube, which is ready to be treated at 95 degrees C for 5 minutes. And we will use the Qsep100 Mini to do this treatment to create the Lysate. So at this stage, we will transfer the saliva samples, which we already transferred it into a PCR tube and added the Proteinase K. We will transfer it to our Qsep100 Mini and then turn on the machine, and we will insert the pre-programmed key into the machine and then close the door and press the button to start the inactivation for 5 minutes at 95 degrees C. So at this stage of the workflow, we will do the Lysis buffer preparation. So once the samples, the saliva and the Proteinase K mixture have been heated to 95 degrees C for 5 minutes, then they will be cooled down to 4 degrees C, and 20 microliter of the saliva mixture will be transferred to a clean PCR tube. And this is where we will be adding the 0.2 microliter of the RNase inhibitor to 20 microliter of the RNA lysis buffer. And this mixture will be added to these tubes. and they will be incubating for 10 minutes. And then immediately they will be added to PCR master mixes for the next steps. Of course, this video is not going to cover all of these steps. We will have another video to show you all the chemistry preparation and the processing of the workflow at this stage for the PCR master mixes. So at this stage, we have programmed the key for the Qamp Mini for the one-step PCR, RT-PCR. And basically for the reverse transcription, there is a one cycle at 50 degrees C for 15 minutes. And then we have activation for one cycle for 95 degrees C, 10 minutes. And then we will do the denaturation, annealing, and extension for 44 cycles, which will be 95 degrees C, 60 degrees C, and 72 degrees C with its corresponding seconds. And it is the previous slide that you saw. And then we will do another extension for one cycle, 72 degrees C for 5 minutes, and then we will hold the whole thing for 4 degrees C. So the pre-programmed key will be inserted and the samples will be transferred. All the 8 samples, which we have several saliva samples, treated saliva samples. Then we have positive control, and then we have few non-template controls, which will be inserted to the 8 wells of the Qamp Mini thermal cycler for the next step of the RT-PCR. Close the door and then press the switch to finish the process in about 1.5 hours. So once the cartridge has been inserted into the Qsep100, we will latch it first. And the latching basically brings all the pneumatic, electrical, and optical connections to the cartridge. Sample tray has been loaded with the sample and with the buffers. And now we will move it to position, part position. And as we set up the system for the 8 sample runs, we will choose a method for this particular cartridge, which is an S1 cartridge. We will choose the 8 kV 200 runs, and I'll show it on the following slide how to do this. We will name it, whatever it is, the name of the run. And then we will hit the run to start the process. Which will again take between half an hour to 40 minutes for the 8 sample runs. And once it's finished, I will show you the results. So at this stage, we have chosen the 8 sample runs, which we have transferred all of the 8 samples, individual PCR wells from the QAMP Mini into the Qsep100 system. We have inserted the S1 cartridge, we latched it, and we will be running it at 8 kV 200 seconds. And I will also name this run COVID demo, and then we will hit the run, and automatically the instrument will take over, and it will go through each individual purging, cleaning, injection of the lower and upper markers, and then individual sample injections, and then it will display results. So besides the 8 samples, the PCR samples, we also use a DNA ladder run, so there will be total of 9 DNA ladders, 9 sample runs, including a DNA ladder run. So what the system is doing right now, it's in a high voltage park position, and for 66 seconds it will be doing a purge, introducing a fresh gel from the cartridge reservoir into the capillary, and then once the 60-second is over, then it will go into the clean position, and it will do a double dip into the clean water, and then once that has been completed, then it will go into the separation buffer, and it will do a separation. So right now, again, the gray one is the current, total current, which is showing around 13.92 microamps of current, and raw counts is around 11,000 raw counts, and the time finished, the 60-second high voltage purge finished. Now it's going into double dip cleaning, and then once that is completed, then it will inject the alignment marker, which is a lower and upper marker of 20 base pair and 1000 base pair. And once that has been injected, which is basically about a 10 seconds for the MA1 position, you can again see the current 14.50 microamps. Now it's going into the DNA ladder well at MA2, and I also will do a 10-second injection. So again, the red is the baseline, red line is the baseline, which is measured in relative fluorescence units, and the gray one is the current, which will be removed once all the electrophoregram results have been accomplished. So now the cartridge is in the separation buffer, and now we are displaying the actual separation results. So again, the red line is the baseline noise with the current being displayed in gray color. So the actual individual peaks will be showing up a little bit over 1 minute. So now you can see we are basically decrementing the time in seconds, 165 seconds, and the total current is 35 microamps. So it will go automatically through the whole process of analyzing all of the samples, including the DNA ladder, and in the end it will show all the results in electrophoregram or gel view, and then I will show you the report for all of the sample results. So very soon you will see here the lower marker of 20 base pair is coming out, followed by the DNA ladder peaks, and so on. So this whole thing will finish up in about close to about 2.5–3 minutes. And then it will go to the PCR sample wells and do the same thing, and then we will have the results. So when the run finishes, then it will display the electrophoregram result. This is the DNA ladder run, and as you can see, 20, 1000, the lower marker, upper marker, including all of the peaks for the PBR322 digest DNA ladder, and then followed by the rest of the PCR product analysis for the 8 samples. Okay, so this is what we are expecting in regards to the results for the primer design that we have. We will show the E gene, the N gene, and also we have an internal control which is RNase P at 170 base pair. So if we look at some of our data, you can see in this one here the non-template control is red, and then we see the blue, the same non-template control, and then the 2 positive controls which have the internal control of RNase P and E gene and N gene. So in another result, you can see again red non-template control, positive control here in blue color, E, N, and internal control RNase P, with few saliva samples going from light green all the way to the dark green. And we also have results, pooling results, going for samples all the way A to J. Again, on top, we will have the positive control in red with internal control and an E gene. We also display these results in gel image format besides the electrophoregrams, you can see here. And then another view showing a gel image format for multiple sample runs done in Qsep100, utilizing S1 gel cartridge. But we also can do the same thing for our other products like Qsep1, Qsep400, and Qsep1 Lite. Qsep1 being the portable unit, and Qsep400 is the 4-channel high throughput system.